Hello, everyone. This is Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. And we're joined here today by Burkhard Steuernagel from the John Innes Center in the UK. So I'd like to give you just a brief presentation before we move into the actual webinar. Next slide, please. So we've been having some technical difficulties today, but so the International Wheat Genome Sequencing Consortium consists of over 3,000 members in 70 countries. We have 8 sponsors, and we work with over 800 research institutes. So I hope we are, yes, so next slide, please. We are able to have this webinar because of our sponsors. So we have 8 sponsors, and they've been strongly supporting us for a number of years, and we really appreciate their contributions. If you'd like to be a sponsor or to get more engaged, please let us know. Next slide, please. Going backwards. Yeah, next slide. So our vision, what we call IWGSC 2.0, is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And our presentation today will get into, specifically into some of the activities that are going on in that regard. Next slide, please. So we are having a delay in the slides, unfortunately. So very quickly, our 2020 activities, even though they've been slowed down due to COVID, we are expanding our Arbor Biosciences collaboration. We are trying to get RefSeq 2.0 published, to get it released. We have been increasing our annotation, both manual and functional annotation, and preparing a version 2.1, which will be available early next year. We're trying to get the funding to do the sequences of 8 land races, and then we're also always trying to get access to the genome sequences for elite wheat varieties and other genomic resources, and to bring those into availability for the entire community. And then finally, we have the IWGSC Webinar series, which we began in January of this year. Next slide, please. So just to let you know, our next webinar will be on October the 7th, and Edward Akinoff from Kansas State University will be giving us a talk about his most recently published paper on differential chromatin accessibility map. Next slide. Just so everyone knows that the presentation will be is being recorded and will be posted on the IWGSC YouTube channel in a few days. You can subscribe to the channel so that you never miss one of our uploads. The presentation will be followed by Q&A, and if you have questions, please submit those to the Q&A panel. Do not use the chat button. You can use the chat button to talk with fellow attendees or to reach out directly to the organizers. And you have 2 handouts, both my presentation and Burkhard's presentation are available for you to download at this time. Next slide, please. So with that, I'd like to turn it over to Burkhard, who will talk to us about resistance gene cloning in wheat. So thank you, Burkhard, for joining us, and let me turn it over to you.
Can you see my screen, Kelly?
Yes, we're good to go.
Okay, thank you. Good afternoon or good morning, or wherever you are, maybe good night. Welcome to this talk. I want to talk a little bit about resistance gene cloning in wheat. So plants have diseases, and quite often these diseases are caused by pathogens. Now, if these plants are our crops, we don't like them having diseases because that causes yield loss. What can we do against that? Of course, the farmers can spray against the pathogens, but we can also try to breed crops that are resistant against the diseases in the first place. So in these breeding efforts, it's very convenient if we know the sequences of the genes that are underlying these resistances. So today I want to talk a little bit about the annotation of one of the major disease resistance gene families, the NLR genes. And then I talk about mutational genomic pipelines to clone resistance genes. And I also want to talk a little bit about AgRenSeq, which is another method how you can clone resistance genes. So let's start with the NLR gene family. This is a large gene family that encodes intracellular receptors, which have a nucleotide binding site. This is this NBARC domain, and they also have leucine-rich repeats. So typically we have several hundreds of these genes in a plant genome. So here you can see a little selection of genomes that I just screened for the number of NLRs. But this is, of course, a wheat talk, so these genomes are of less importance here. So let's have a little bit of a more wheat-centric view, and this is also a historical view, going into the assemblies that we had for wheat. So in 2014, we had the IWGSC assembly that was based on the flow-sorted chromosomes. And then there was the assembly by Chapman et al. That was not Chinese spring, that was a synthetic wheat. And then we had this massive jump in quality of assemblies, first by Clavijo et al., the assembly by Earlham Institute, and then we had the Mazurka assembly by Simen et al. And also, of course, we have the chromosome scale assembly, the RefSeq 1.0 by IWGSC. And if you look at the NLRs that can be annotated in these assemblies, we have to distinguish a bit between full-length NLRs and fragmented. So full-length in this case doesn't necessarily mean these are the only functional ones, but this means that we have a full nucleotide binding site and leucine-rich repeats. And the fragmented are just signatures of either of these little domains. And you can see in the old assemblies, you have a massive amount of little signatures or of partial NLR signatures in there. And then when the assemblies improved, we suddenly have far more full-length NLRs and less of the not complete or of NLRs that do not have leucine-rich repeats. So if we go a bit further, this is now a representation of all the NLRs that are in the RefSeq 1.0. These are the confirmed NLRs. I don't want to go too much into detail of this figure, but what you can see here, the blue parts here, these are all signatures that belong to the NBARC domain, and the green parts belong to signatures of LRRs. You can see in the LRRs the massive diversity of copy number and so on. And for people that are interested in NLRs, these colorful parts, the red ones or down here, the other colors, these are integrated domains of NLRs in wheat. So how can we annotate NLR genes? So obviously you could use tools like HMM-Scan to screen for the entire domains here. But there's another option, which is you can use little motifs. So if you go into the protein sequences of NLRs, there are a lot of short, highly conserved motifs. The first set is this one down here. That was defined already in 1999 by Myers et al. This was based on NLRs in Arabidopsis. And then in 2012, there was another set of motifs that have been defined by Yupe et al. It's the set here above. And you can use tools like the motif alignment search tool, MAST, to screen protein sequences for these little motifs. And if you know a little bit about NLR annotation, you can look into the correct order of these motifs. If you know some of them are there and they are in the right order, you could deduce this is most likely an NLR gene. So this way of annotation means that you have to visualize every single protein sequence, which is a lot of work if you want to screen an entire an entire gene annotation. So therefore, in 2015, we developed a little tool that takes the text output of this MAST program and evaluates all these little motifs that are in there and the combinations of them, and then decides if this sequence is an NLR or not. So this NLR parser is a classification of sequences if they are NLR or not. So we can use this to screen protein sequences, but already in NLR parser, we included an option for 6' translation. That means we could also use that to annotate DNA sequences. So this was in 2015, and that was good enough, because if you remember these old assemblies, they were very fragmented. So all of these contigs were rather short, and then suddenly we had this big jump in the quality of genome assembly. Suddenly we had megabase contigs, and this poor tool, this would still annotate things, but if you give it a megabase sequence, it will tell you yes, it is an NLR, or no, which is not good enough. What we actually want on this large contig, we want to know where exactly the NLR genes are. So for that, we developed another tool called the NLR annotator, and this now takes large DNA sequences up to chromosome scale and annotates all the loci on that chromosome that are potentially NLR genes. So just to explain that a little bit more, this is a screenshot here from one NLR locus. Actually, what you see here in blue, this is a gene from the IWGSC RefSeq 1.1 gene annotation, and the little bit bigger parts here, these are the exons. So the NLR annotator first finds these motifs in here, and then it will evaluate the motifs and looks for the right combinations of different motifs that have to be present to call it an NLR. And in that case, it would call the NLR locus from here to here. This motif here, you can see it's already in the intron, but also if you look at the whole context, with a bit of practice, you would know this does not really belong at this position. So NLR annotator will exclude this. One point I want to make here is that NLR annotator will annotate loci. So it will not provide the entire gene sequence. Also, it will not tell you the start of the gene or the end of the gene or the intron exon structure. For that, you would need a proper gene annotation. So you need RNA-seq data to do that. And that gets very quickly complicated. NLR annotator will just tell you where the NLR potentially is, which from a gene cloning perspective point of view, this is the first important thing that you actually need. And then in the second step, you could determine the gene structure. So with NLR annotator, we don't get a gene structure. What we also don't get is a differentiation between genes and pseudogenes. So with this just motif-based annotation, we cannot say whether an NLR locus actually overlaps with a functional gene. But again, from a gene cloning point of view, this is probably even advantageous, because usually when I clone resistance genes, they tend to not be in that reference sequence. They tend to not be in Chinese spring. They are in one of these other accessions, or even in one of the awkward wild relatives. So, but very often what we find in Chinese spring is a non-functional allele. So this is very often a pseudogene. So if we would use a normal gene annotation, to find sequence resources for our gene cloning, this wouldn't be good enough. But if we take every locus that is associated, it's very likely that we find good sequence information. Just to show one thing we can do now with NLR Annotator, this is a little exercise we did together with Simon Krattinger. So he went through literature and pulled out all the leaf rust and stem rust resistance genes that have been mapped. So mapped in this case means we haven't actually necessarily cloned the gene. We don't know the sequence yet, but what we do have are markers that delimit an interval in which that resistance gene most likely is. So what you can see on this picture is for every red bar here, this is for an NLR locus, and the gray area are the regions that are delimited by the markers for each of the resistance genes. And in quite a few cases, we had a large region that was delimited by markers. But if we looked at how many NLR loci we actually had in there, that was less than a handful candidates. So with this information now, you can immediately go into a candidate approach for these genes. So, but that was just a sort of side project. The reason why I'm really interested in NLR annotation and why I needed this tool, NLR Annotator, is resistance gene enrichment sequencing, or short RenSeq. So RenSeq was developed by these two guys down here, Florian Jupp and Kamil Witek in Jonathan Jones' group. And essentially it's the same as exome capture. So if you're interested in resistance genes, you do not want to sequence the entire genome. You don't even want to sequence the entire gene space. You just want this very small fraction that are the resistance genes. So it's like exome capture, which means we would design these little bait sequences that are complementary to the actual NLR genes. So with these baits, then they will hybridize during the enrichment process with the fragments in the sequencing library that belong to NLRs. And the interesting part here is that these baits will hybridize with sequences even if you don't have absolute identity. So this means I can go into all the available references, such as the RefSeq 1.0. I can annotate the NLRs, I can pull out the sequences and design my baits from that. And I can use that and go into wild relatives or into other wheat accessions and do RenSeq, so I can pull out the NLR sequences that are almost the same. So what do we use RenSeq for? First of all, the RenSeq libraries we have right now. This slide is not really for the presentation. This is more, you can download the presentation from the handouts. If you're interested in RenSeq, this is where you find all the sequences for the libraries we have. We have now version 3 for Triticea RenSeq library, and we have an Aegilops tauschii specific RenSeq library. And the last link is where you can actually order the enrichment kits. So what do we need RenSeq for? One concept that we use for cloning resistance gene is mutational genomics. So just to show how that works, if you have a bag of seeds from a resistant accession and you know that this resistance is conferred by a single dominant gene, you can do an EMS mutagenesis screen. That means we treat the seeds with a mutagen that induces mainly point mutations. So then we need to go into the next generation to actually fix the mutants, the mutations in the mutants, and then we can challenge the mutants with a pathogen. And with a bit of luck, this mutagenesis has rendered some of the mutants susceptible. So they have lost the resistance, or it means they must have a mutation right in the resistance gene. So if we pull out these susceptible mutants, we can do the mutational genomics. So essentially, if we sequence or line up all these resistance genes and compare the sequence of the mutants to it, we will find all these random mutations in the mutants that are induced by EMS. But since we selected for loss of function in this one very resistance gene, we would expect that in one of these resistance genes, every mutant must have a mutation. So in this case, it's obvious that would be R7. So that would be our candidate. So I already said we combine this mutational genomics with RenSeq. So the pipeline we call MutRenSeq. And essentially we do this EMS mutagenesis screen, and then we do RenSeq on the wild type and on the susceptible mutants. And then we run a de novo assembly on the wild type RenSeq data. So this means we create our own reference on the fly. We don't need to rely on any external reference. We create a reference on our own wild type on the fly. So this is RenSeq, which means this is sequence enrichment. In that assembly, not all of the contigs we have are actually NLRs, so we need to annotate again. This is again NLR annotator. And then we use this as a reference to map the data from our susceptible mutant RenSeq data against it. And then the mapping and the contig annotation is fed into a program called Mutant Hunter, and this does the actual mutational genomics. So for each NLR associated contig, it checks how many of the mutants have a mutation in this contig. Just to emphasize that again, so this is again a screenshot from a genome browser. And what you can see here in the different tracks is the lowest track are the familiar motifs. So this tells us this contig is actually associated with an NLR. And then the first track here, this is our wild type. We don't expect any SNPs in here. If we find any, this is a sign that we have a problem in our de novo assembly. And then for all of these other tracks here, these are for the independent mutants. And the more mutants that have a mutation, the more likely this is our candidate. So in this case, each of the mutants have a mutation, and therefore we assume this is our candidate. And usually this is quite clear. The first time we tested that was on material for a resistance gene that had already been cloned before. That was SR33. That was a stem rust resistance gene. And when we run this MutRenSeq, we got a single candidate out of that. So that was very encouraging. And we looked for additional targets. And the first thing we found was SR22. So Sambasivam Periyanan in Evans Laguda's group, he was trying to clone this SR22, and he was working on this for quite some time already, because he had already produced 6 EMS mutants. So the gene was introgressed originally from Triticum boeticum into hexaploid wheat, but it was a bit problematic to clone that with traditional map-based cloning because it was in an area of suppressed recombination. So just to show a bit about SR22, this is how a leaf looks like when it's infected with stem rust in the and the genome has SR22 present, and this is when SR22 is missing. Similarly, this is the wild type that was used for the EMS screen, and these are the susceptible mutants when they are challenged with stem rust. So we ran our MutRenSeq pipeline here and again got a single candidate gene. So this is this was the first novel gene, so it had to be validated. And we did that by transforming the cultivar fielder. This is susceptible against the stem rust race we used here. And when we transformed, then we got some transgenics that had regained resistance. So this was the proof that SR22, or our candidate gene, was actually SR22. So that was very, very nice result. And we published that. And at the same time, we also worked on another gene together with Evans' group. That was SR45. So that was essentially the same story. They already had susceptible mutants. We ran MutRenSeq, got a single candidate, and got the transgenics. So we had cloned another gene. So this is MutRenSeq that works very nicely. Although the problem is with this method, you run it, you get a single candidate, everything's great. You open the champagne and everyone's happy. But every now and then, you don't get a candidate. And in those cases, the whole experiment is pretty much inconclusive. You don't know what really happened. Was it a biological reason? Was it a technical problem? In most cases, we would assume that the RenSeq, so the bait library that I use for enrichment, was not good enough. So I couldn't enrich for the target gene. So if the target gene is not sequenced, we would never see anything in a MutRenSeq experiment. So this could obviously be because the bait library isn't good enough. I mean, there's the reason that we are already in version 3. We constantly improve the bait libraries. But it could also be that that the target resistance gene is simply not an NLR gene. This is a large gene family, but there are others out there. So what would we do then? We could use exome capture, but again, it could be that we simply don't enrich for the gene. So the option we went for, we called MutChromSeq. And here we teamed up with Jaroslav Dolezal, and most of you probably know him because there's a long history with IWGSC and Jaroslav. I mean, he did the flow sorting for the chromosomes for the 2014 assembly. So if you think about our mutational genomics and why we used RenSeq, we didn't want to sequence whole wheat genomes. So that's just too much. We wanted this complexity reduction, which was really great with RenSeq. But we always had this assumption, it needs to be an NLR. So in this case, we just need to know on which chromosome our resistance gene is. And if we flow sort and just sequence the chromosomes we're after, we have a 21-fold reduction in complexity, which is also very nice. So our first test case, which we wanted to use as a proof of concept, was not in wheat. So this was in barley, and the gene was called Aceriferium Q or SIRQ. So this was worked together with Nikolai Adamski. This gene is not an NLR, is not even involved in resistance. This is involved in glaucousness. So the mutants, the loss of function mutants, are called Wechsler's mutants. So they are historical mutants. We could order them from a gene bank. And when we ran MutChromSeq, we got a single candidate. So it worked very well. Luckily, or maybe unfortunately, there was another group also working on the same gene, and they map-base cloned the gene. So we had immediately a confirmation that we had the right gene. Unfortunately, they also published. But more importantly, this was just a barley gene. And at that time, I already knew in a couple of years I would have to give this IWGSC webinar. And you can't just present a barley gene in a wheat seminar. So I really needed a wheat gene. Luckily, at that time, Beat Keller visited us at JSC, and he told us that Javi in his group, he was working on PM2. And they had pretty much the same idea that we had. They also collaborated with Jaroslav, and they had already flow sorted wild type and susceptible mutants for PM2, and they had even sequenced that. So they were very kind, giving me the data that they had produced. I ran it through my pipeline, and we cloned PM2 with that. So here we have the wheat gene, so I can present it in the IWGSC seminar. Okay, so we have two very nice pipelines that work on mutational genomics. This is essentially forward genetics to clone single dominant genes. And well, we've been successful with these methods quite often, but there's always the things you need to consider. Each of these experiments is to pursue a single gene. So that means in most cases, we need an extra EMS mutagenesis screen for each gene. And that is a lot of work because the EMS mutagenesis itself, that's not a trivial experiment. When you do that, you usually need to find the sweet spot between mutating the plant enough that you have a chance to get loss of function mutants. But if you mutate the plant too much, you simply kill it. In hexaploid wheat, this works all right because you have some redundancy in the genome, so you can hit it quite hard with EMS. In diploid relatives, it is really, really difficult. So I would also like to introduce another method that we use for cloning genes, and this is called AgRenSeq, association genetics using RenSeq. So this is work I did together with Sanu Arora, and we used a wild diversity panel of Aegilops tauschii. Aegilops tauschii is a diploid, and it's the D genome progenitor, so it's closely related to wheat. And we again were after stem rust resistance genes. So Sanu assembled this diversity panel of different Aegilops tauschii accessions, and each of these accessions were phenotyped with several stem rust races. And we also did RenSeq on each of these accessions. So again, we want to sequence a lot of accessions here, so we need a complexity reduction. So when we have all this data, we want to do association genetics. Traditional association genetics would be based on SNPs. Now, if we want to use SNPs together with NLRs, it gets really difficult because between accessions, if you look at the NLR complements, you have a massive variation in copy number and in presence absence. So SNP data is almost impossible to use here. Instead, what we used was something that is called K-mers. So K-mers are little sequence signatures, and you can use them almost similar to SNPs. The data is a bit noisier, but on the other hand, they do not only represent single nucleotide polymorphism, but they also represent indels. So when we run AgRenSeq on such a data set, we get plots that look a bit like that. So this is very, very noisy or busy. So let me talk you through this one. On the x-axis here, you have lined up all the contigs from a de novo assembly of a resistant accession. So here we have all the NLR contigs. If you imagine it, it's essentially like lining up all those R genes. So on top of each of these NLR contigs, we have a column of dots here. And these dots represent K-mers. And the height of the dot, or where this dot is on the y-axis, is the association score. So the higher the association score, the higher the dot is on this y-axis. And the bigger the dot is, the more K-mers in that very NLR contig have this association score. So again, this picture looks very, very busy. But if we zoom into the top a bit, we can see there's a clear candidate. So in this experiment, we screened the Aegilops tauschii panel with a stem rust race called RKQQC. So special about RKQQC is there's a known gene that confers resistance against that, and that is our good old friend SR33 that has already been the proof of concept for MutRenSeq. So behind this dot here, that's the winner with the highest score, are 87 K-mers. And if we look at the NLR contig that contains these 87 K-mers and compare that to the coding sequence of SR33, we see that the contig contains all the exons, obviously also then the introns, but we can also get a bit of upstream sequence and a bit of downstream sequence. So just imagine that we had this panel, we phenotyped, we sequenced, run our pipeline, and just like that, we have a complete candidate gene. So we could almost take this sequence into a construct and transform it immediately. We would probably need a bit more of promoter, but it is a very quick way to find candidate genes. So this was really an exciting result. But we had also screened the panel with additional stem rust races. So the next race we looked at was TRTTF, and we again have a very clear winner here. So Sanu looked at this sequence and also did a little bit of literature research and found out that a very likely candidate for this one would be SR46. That would sort of fit. And we had heard that our collaborator Evans was also working on SR46. So we sent him the sequence and asked him if it looked familiar. And we got the excited email back that it indeed looked very familiar. So we very quickly got confirmation that we had cloned our first novel gene here with AgRenSeq method. And then we moved on, took the next stem rust race and found another gene called SRTA1662. So this was pretty much when I left the group of Brande Wolf, where we did all these experiments. Sanu and Kuma obviously continued with AgRenSeq and worked more on it. They cloned additional genes, and they also improved the method. They added a general linear model to cope with the population structure of our panel. So now the plots that we can generate look much, much cleaner than before. So, but just remember, you have an AgRenSeq diversity panel, and you can just plug in a phenotype. You can screen this panel with a pathogen, and we can run our pipeline and immediately get candidates. And the good thing is that we have now established this panel, and it is sequenced. So the only thing that has to be done now is use an additional pathogen to screen it, come up with a new phenotype, and we get additional candidates out of this panel without additional sequencing. So to summarize what I was talking about today, I first talked about NLR annotator, which is a tool to annotate NLR loci, not only in wheat, but in all plants. I talked about mutational genomics, if you want to pursue single dominant genes. And then in that context, I introduced the complexity reductions, RenSeq and chromosome flow sorting for mutational genomics. And then I talked a little bit about AgRenSeq. So with that, I'm almost done. A big thanks to Brande Wolf. All of this work was in his group. So I also would like to acknowledge Inma, who did the first RenSeq experiment on Triticea. That was the one we could then use to clone SR22. Sanu Aroa worked on AgRenSeq. Sam Perianan cloned with us SR22 and SR45 in Evans' group. And Evans also worked with us on AgRenSeq. Florian and Camille in Jonathan Jones' group invented RenSeq. Nikolai Adamski, Javi Sanchez and Beat Keller for the MutChromSeq. Jaroslav and his team did the chromosome floor sorting. A big thanks to Jake Eng from Arbor Bioscience. They supported us massively when we did our AgRenSeq study. Brian Stephenson did all the phenotyping for AgRenSeq, and Jesse Poland contributed with a large number of Tauschi accessions for the diversity panel. So with that, thank you for listening, and I'm ready to take some questions.
Great. Thank you very much, Burkhard. We do have a number of questions. Just to remind everyone, please insert your questions in the Q&A panel on your dashboard for the webinar. Do not put it in the chat, as we will not be using that. So the first question is, for the EMS mutant, what's the percentage of EMS type variation? Should it be greater than 90%? And if it's only a small part, say 30%, do we assume that there's some contamination, but not a real mutant?
It's not completely easy to answer. So I assume this goes into the direction if we assume the SNPs to be the canonical EMS mutations. If we see others, this could come from contamination. So just the experiment didn't work. We have other introgressions there. It could also come from problems during the bioinformatics. If we have problems with our assembly, we will see a lot of SNPs there. it's difficult to answer that. You probably always need to look into detail.
Great, thank you. So the next question is it a good idea to use the IWGSC RefSeq version 1.0 as the reference to identify resistance genes in progenitor species, particularly if you have mapping populations or mapping information available for those?
I would say the IWGSC RefSeq 1.0 is the best sequence we have at the moment. That said, it can always be that a resistance gene you're after is just not present in Chinese spring. So that's always a thing to consider. The first try would always be use the Chinese spring sequence. And if that doesn't work, we need to go a bit further and employ other genome sequences that are out there. I mean, there are 15 other genomes of wheat published already, and they are also nice sources for sequence information. But I think in most cases, the starting point is still our Chinese spring sequence.
Great, thank you. Is it possible to clone a gene of interest other than a resistance gene using the MutRenSeq techniques? And I think you discussed that during your presentation, but could you elaborate on?
With MutRenSeq, it's not possible because RenSeq simply enriches only for NLRs. It is obviously possible to design bait libraries for other gene families. I mean, you could design a bait libraries for RLPs, RLKs, or something completely not involved in resistance. MutChromSeq works very well on genes that have nothing to do with resistance. You need a clear phenotype, and it needs to be this single dominant gene. Otherwise, the EMS mutagenesis screen won't work. But if you have loss of function mutants, MutChromSeq is a great approach.
All right, great. When you de novo assemble the MutRenSeq or RenSeq sequencing data, don't you get a lot of false chimeric contigs?
Yes.
Okay.
And this can massively be a problem in the experiment. I assume that some of the experiments where we did not get a candidate are because of that. We have been working a lot on trying to improve the assemblies, and I think there are still experiments running in Brandes' group. It worked a lot of times.
Okay.
So there are problems in the assembly, but you can adjust that a bit by being more relaxed about the allele frequency of SNPs you are after.
Okay. So what sequencing technology and assembly tools are you using?
Sequencing technology, all what I presented today was based on normal Illumina sequencing. It is possible to do RenSeq using PacBio. We did not have good results with that simply because the coverage of the PacBio sequences wasn't good enough. So I did a MutRenSeq experiment and I had PacBio data on my wild type. And I also had Illumina data, but I only used the PacBio data to do my de novo assembly. And I didn't get a candidate. But when I then used the standard way of doing a Illumina de novo assembly, I suddenly got my candidate. And I figured out that my candidate sequence was simply not present in the PacBio reads. So at that point, we stopped using PacBio for our experiments. But obviously now the technology has improved a lot. I think the second part of the question was about the tools, or?
Yeah, the assembly tools, yes.
So our assemblies were done with a CLC assembly cell. We tried Mazurka in between, which also works nicely. essentially you just need to get an assembly. And I think the tools improve constantly. Since CLC worked nicely for us, I never went away from that one.
Okay, great. So on slide 5, you showed the data that you showed illustrated a drop in the number of NLRs in the recent assemblies. Do you have a reason for that?
Not really. It was just a way of introducing the annotation of NLRs. So I used my NLR notation a bit as a benchmark for the assemblies. And obviously, whenever a new assembly came out, I was interested how much more sequence information or NLR information would be in there that I could push into my bait libraries.
Okay, great. So what about SR13, which is one of the important genes in durum? It is, and it's also found in Fielder.
I don't believe it's cloned.
No, I haven't seen anything, but
I don't know. If somebody wants to do a mutant screen for that, I would be very interested in working with the data.
right. So how do you control for misassemblies during your assembly process, or did you perform any quality control steps after that?
No, I don't have quality control. You will see, so the main quality control that I have in the experiment is mapping the wild type data against its own assembly. So there, if there's a chimeric contig, if something collapsed, you would see a SNP frequency there. So I think currently in the pipeline, what happens is if you have some sort of weird allele frequency in the wild type mapping, this position gets excluded from downstream analysis. So that would, in the worst case, mean if one of the mutants has a mutation right at that position, we wouldn't pick this mutation up. And then our candidate gene would have one less mutant to support it.
All right. Can you talk a little bit more about mutations downstream of the R genes that will not be seen in RenSeq? And what to do about that?
We can't do anything about them. So I guess what could be is that in the downstream signaling, something is so severely disrupted by the mutagenesis that we have a loss of function mutant or something that looks like that, but we don't see any mutation in the resistance gene itself. That would produce an inconclusive experiment for us.
All right. So what's the optimum concentration of EMS used for mutagenesis?
I have no idea. I'm a bioinformatician.
Yeah.
I can ask around and send an email to that question.
All right. So are you aware if the method is being used for other gene families and traits? Perhaps modified?
Our method.
Yeah.
I know there is a bait library for RLPs, RLKs out there, so that would be a modification of MutRenSeq. And obviously, I already said that MutChromSeq can be used for any other gene and has been used.
Right. So can you comment on the cost? And perhaps you can't, since this was done in Yaroslav's lab, and the difficulty of chromosome flow sorting. And
So I
perhaps
think
this person is asking, one, how much it costs to have it done, or secondly, whether they can actually do it.
I think prices constantly change there. So I can't say very specific numbers, just I guess to give a ballpark figure, and I hope I'm not completely wrong, is that a MutRenSeq experiment, just the sequencing and so on, costs less than 10,000, whereas MutChromSeq, first of all, you need Yaroslav, otherwise it doesn't work.
Right.
And you now have to sequence chromosomes, so that is much more sequence data you have to produce. But again, I might be completely wrong because sequence costs decrease constantly.
Yep. So can you use the pipelines with transcriptome data instead of whole genome?
I've tried that once and was not successful with that specific experiment. But again, later when that gene was cloned, I didn't find the gene itself in transcript data. So if the gene is properly expressed, in theory, it should work. But I don't have a proof of concept on that.
All right.
I would always try to work with genomic data in this context because it's just so much easier to deal with than transcriptome data.
So what do you do if there's a situation where a trait is not explained by a single gene and you know that there's probably more than one candidate gene? Is there a process that you use, or how would you approach that?
So I guess all the mutational genomics approaches won't work because it's very unlikely to get a proper loss of function mutant if you, if you knock out one of them. I suppose if it would be enough to get a loss of function if either of the genes is knocked out, then it could work if you have enough mutants that just randomly support both genes, but it gets much, much more difficult.
Okay. So how does DART-seq genotyping work for AgRenSeq? Or are you aware?
I think I've never used DART-seq data. I have no idea if you can use that.
Okay. So is it possible to identify multiple major NLR genes in a panel using AgRenSeq if the panel has been phenotyped using several pathogen races?
Mm-hmm. Well, that's exactly what we did. So first of all, if you have several resistance genes against the same race, now that it's adjusted for population structure, I think I showed this picture at the end. Too far. Where are we? Here. So when I first looked at this data without the GLM, I would only see the SR46. But as soon as you adjust for the population structure, you also see another resistance gene coming up. This is SR45. So even with the same race, you would potentially see several genes. But the other thing that we did is you screen the panel with the first race and you find a resistance gene. You screen it with the next race, you find another one, and so on and so on. Just the same way as you can use not only several races, you can also use other pathogens to find other resistance genes.
All right. What is the best strategy to apply when the candidate gene is located on an alien translocated fragment?
So I guess mutational genomics, or well, if we use MutRenSeq, it doesn't care where that gene is. if it's on alien translocated fragments, we would still see it, because we have the wild type de novo assembly, which would include every NLR that we can capture. So that would be no problem. If we do chromosome flow sorting, we need to be very certain on which chromosome it is. So we would have to have genetics on that including that very accession where we clone the gene from. But again, there we should be able to find it despite an alien introgression. The good thing is with the mutational genomics, it doesn't rely on genetics. So when we do map-based cloning, which is based on genetics, the introgression will likely cause problems with the recombination, so you don't get close. But with the mutational genomics, that's not an issue.
All right, the next question is, have breeder-friendly markers been developed for each of these genes that you have cloned? And if so, where are they being used in resistance breeding in wheat?
I don't actually know. I should know that. But the sequences are published, so it's very straightforward to design your own markers on those. So now that you have the gene sequence, you can design the perfect marker.
All right, the next question is, how many strong functional motifs identified which distinguish TIR, NBS, and LRR regions of our genes, along with other signature sequences like WRKY?
So we use this set of 20 motifs that has been defined by Juppe et al. And you can see here, this is a canonical NLR. So these motifs would always be present if you have a standard NBARC domain. And this 1716, these two motifs show a coil-coil domain. Sometimes the 17 will not be recognized that well. There are 3 motifs that very clearly show a tier domain if it's present. So if you work on dicots, you will see those. In the tier domain, as soon as you look into the NBARC domain, this RNBSA or motif 6 is missing. And then we have 3 different motifs that signify these leucine-rich repeats. And these can come up in different combinations and copy numbers. I hope this answers the question.
Well, Burkhard, I want to thank you very much for a great presentation and your efforts to answer these questions. It's a great tech and great forward progress for wheat. And just to remind everyone, the presentation has been recorded, and it will be available on the IWGSC YouTube channel, so be sure and keep an eye out for that. And it is free to share. So thank you again, and we look forward to seeing you on the 7th of October for our next webinar series. We hope everyone has a great day.